Hi, I'm Jason Darell, and I'll be presenting on the work I did over the summer in the Department of Anatomy and Structural Biology at the Albert Einstein College of Medicine under the leadership of my mentor, Dr. Weili Liu. So first, to give you some background, my lab focuses on transcription, which is the first step in the creation of a protein. It's when the DNA is copied into RNA, which then travels outside into the cytoplasm of the cell to the structure, which creates the protein. And transcription is made up of 3 steps: initiation, elongation, and termination. And my lab mostly studies initiation, which is the step in which all of the proteins bind to the DNA to commence the transcription process. So what first happens is this large protein, TF2D, binds to the DNA, which in this case is a piece called BAX. This then attracts all of these other proteins, including P53, which is an activator. P53 is a tumor suppressor, which means that it helps the body, it helps prevent cancer. So our main goal is to create a 3D model of this entire complex, but that's very complicated. So for this experiment, we're only using these 4 elements: TF2D, TF2A, P53, and DNA. So we purify each of these 4 things separately, freeze them in vitreous ice, which is ice that's been frozen so fast that it's almost like glass, put in the transmission electron microscope, which passes an electron beam through the sample. This creates a 2D projection, almost like an X-ray, and then go through a series of computational analysis in order to arrive at our final goal, which is the 3D model of the complex. So one of the main things that I was involved in was DNA purification. So we start out with a small piece of the BAX promoter. We run it through the PCR, which amplifies the DNA. We then go through a series of steps, which are called gel band purification. The results, a little bit of the results of this are used to quantify the DNA, while most of it is used in our purified sample to be put in the electron microscope. So this is one of the images taken from the electron microscope. As you can see, it's very low contrast as well as very low resolution, so you can't see very much. As a result from this, we need many of these pictures, almost 20,000, in order to create an accurate 3D model. And what also makes this process difficult is that the particles you pick have to be isolated from all the other ones. Otherwise, the 3D model you create will reflect 2 particles instead of just 1. So this is that same image after the particles have been picked. These large boxes are these red ones zoomed in. And in the middle of each box, you can see a small particle, which is this complex. As I said before, you need 20,000 of these particles in order to create a 3D model. So these are the results we have so far. These are called 2D class averages, which is basically the computer compiles the data of the particles you picked and figures out what the average image of the particle is from a certain angle. So many of these 2D class averages will be used to create the 3D model. So future work is to continue to pick particles for computational analysis, including creating more 2D class averages. And the final goal is to construct the 3D model of this complex. I'd like to thank Dr. Weili Liu, Dr. Anna Piasecka, Mrs. Lihua Song for assisting me in my lab work, and Dr. Rachel Cox for helping me make my presentation. Thank you.